It's amazing that explaining life's immense diversity all comes down to some genetics and some biochemistry. And life on Earth is just one family. And what's true for you is true for all biology.
Hello, welcome to Genetic Shambles. This is, I think it might unofficially be the third in the series, but we've certainly done three. But I think maybe only one of the other ones has been official. So this is brought to you by, amongst other things, well, not amongst other things. In fact, the only people who bring it to you, apart from me and our producer Trent, are the Genetics Society and the Milner Centre for Evolution, which is at the University of Bath. And also, we should mention another podcast that is available, which is Genetics Unzipped by Kat Arney. These podcasts that we're going to be doing are going to be fortnightly, and they're going to be looking at different ideas and different ways that we are currently understanding and researching in the world of genetics. Today, we're going to be looking at some of the I suppose what might seem initially some of the simpler questions in terms of some of the simpler definitions, but from that, of course, a tree of life will grow that will become increasingly complex by the end of this episode. We should also mention Science Shambles Q&A, which is on every Sunday live at 3 p.m. And on that, people can send in questions live. And I can tell you tonight as well, if you're watching now, if you have got questions about genetics, about the human genome, about other genomes as well, then you can ask them, and Trent, our producer, will send them to me. So today's guests are, first of all, Sarah Teichmann, who is Head of Cellular Genetics at Wellcome Sanger Institute, Director of Research, Cavendish Laboratory in Cambridge, co-founder and co-chair of the Human Cell Atlas Organising Committee. And I was watching something about that earlier on, and that alone is utterly fascinating, and I think would probably fill a lot more than an hour. I mean, it's looking at every single cell in the human body, but we haven't fortunately got there yet, so we won't be dealing with every single one of the cells of the human body. We also have Gil McVean, who's the Chief Scientific Officer at Genomics PLC, a visiting professor at the University of Oxford, and we have Adam Rutherford, who, amongst other things, of course, on Radio 4, is the presenter of Inside Science, The Curious Cases of Rutherford and Fry, and has just written an excellent book, which is somewhere on my desk at the moment, which is How to Argue with the Racist. And also, I suppose his most gene-specific book would be Creation, which is a flip-up book, basically, which is, first of all, you read about the history, and then you read about the future, and you can just flip it over, and they are done in reverse. So it is a lot of fun to read that book. Well, I mean, there's one bit of fun. You turn the book over, and then it's just more reading about stuff, But we are... I'm being very flippant today, by the way, because I realise that this is not going to be a flippant podcast whatsoever. But I will tell you now that we will get, actually, I think, possibly later on to some of the discussion about genetics, which is in How to Argue with the Racist, because it is some of the misunderstandings remain pertinent today and are still things which are thrown up a great deal. But I'm going to start off. Sarah, if it's okay, I'd like to start with you, and in fact with everyone. Please feel free to join in at times with the answers, which is, we should start off with definitions. And it's nearly 20 years now, isn't it, since the mapping of the human genome. So can you tell us what is a genome? What does that actually mean?
It's the nucleic acid content that's inside every single cell, and carries the information that encodes the organism, basically the cells and molecules in the organism.
And it's basically, and so this is, I was talking with Paul Nurse the other day, and the thing that I find remarkable is that every single cell within the human body has the potential to remain living. So we could in some ways take every single cell. You could slowly pick apart a human being. How many trillion is it again? I can't remember now. Is it 300 trillion? I may be
37 trillion estimated cells in a human being. Yeah.
I have made a way too big human in my laboratory. I've gone and used 300. Oh, ridiculous! But that seems to be so. In every single cell, basically, we have the information to make that living thing again.
Yeah, that's right. Yeah, in principle, that's correct.
And so I'll ask you, Gil, then. So when we're also a definition of the gene itself. So when we're talking about genes, and I know that one of the things that surprised, I suppose, the researchers when the human genome was mapped, that there were actually fewer genes than was imagined for something which is considered so complex.
Yes, I remember there being a sweepstake about how many genes there'd actually ever be found in the human genome back at the time it was being sequenced, and estimates had been in the more like 100,000 range, but it turned out to be closer to 20,000 or something like that. The idea of a gene and the definition actually go way back, though. It really predates our ability to sequence DNA, or in fact, know anything about DNA at all. And the earliest definitions of genes are really about a point in a sequence, in an organism that can be mapped to a trait, such as flower color or the Mendel's traits on peas that he was studying. So it was to do with inheritance, essentially. It's something that you can follow through the generations. And as our understanding of the molecular basis of those traits grew, so we began to understand, I think, that there's not a one-to-one correspondence, clearly, between the things that we find in the genome and those traits, but there's a sort of proximal one-to-one correspondence in that there are bits of this genome that you can pull out, which encode for small bits of machines, typically proteins, sometimes RNAs, which act as part of this amazing machinery to make the cells that make the tissues, to make the organs that ultimately make the individual. So it's the notion of the genome is really around trying to find these units within this massive sequence of DNA, which are sort of independent in that you can pull them out and they do a function, although often they do many different functions.
Well, is that, Adam, because you've quite often, I've seen you do presentations about misunderstandings, and certainly that's something that you've dealt with on some of the Radio 4 shows, that there is that idea of at one point, the popular press, well, all the press, in fact, were fascinated in the idea of a single gene for a single attribute, weren't they? To say that there is the gene for this, there is the gene for that. How, as Gil was just saying, that idea, though, is not true, is it? That you can't merely remove one gene and say, that's the one that creates this particular color, curl in the hair, or whatever.
Yeah, that's right. So this was a lot to do with the thing you just alluded to, and the thing that Gil just said, which was that betting book, which I've actually got a copy of right here. So this page, I don't know whether you can see that, but this was the page that Ewan Burney, who's the head of the European Bioinformatics Institute now, but he was a PhD student at the time, which was 20 years ago. So the announcement was on June the 26th, 2000, which was Bill Clinton and Tony Blair and Craig Venter, and tell me who the fourth one was, Gil.
All right.
Francis Collins was. Yeah. Crikey, sorry. To all the
ever since you did that alpha course, anyone who attaches themselves to religion, you find it harder and harder to recall their name.
It's a complete
extremely unfair.
Yeah. Anyway, but it was to the idea that even the best geneticists in the world, human geneticists in the world, didn't really have a good grasp of the number of genes that we had, was a lot to do with this very baked-in idea in both science and culture that there is this unit of inheritance, and that is what we call a gene, and that for each characteristic there will be a gene that encodes it. And one of the things I argue in that book is that this is something that emerges out of early genetics, and like Gil was saying, Mendel and his peas, which we learn at school, he had a very one-to-one relationship between a unit of inheritance, which would become known as the gene a few years later, at the beginning of the 20th century, and whatever the characteristic was, like wrinkliness of skin or flower color, whatever. But I think the idea goes way, way much further back in time than that. And the way that we've been thinking about inheritance for literally thousands of years really does reinforce this idea that there is a thing, something which is transmissible in a sort of discrete way from generation to generation, which we now call a gene. And it encodes specific things. So the earliest example I'm aware of is from the Talmud, where there's a very accurate description of an inheritance pattern for a lethal disease, which is effectively the rabbis excusing sons from being circumcised at birth. And because of the pattern of inheritance that they describe, if your first son has died from bleeding at circumcision and your second one is in one cousin, then the next son is exempt from circumcision. And we think that is now haemophilia. So this is an idea that is so baked into both culture and genetics that it truly was a revelation in 2000 when that betting book was done, and everyone was wrong. The highest estimate was out by an order of magnitude. The lowest was still out by about 7,000 genes. And there's a lot of discussion about what a gene actually is, what that definition is, and most of the rest of that page is in fact like proper sciencey caveats to what is a gene. And so I think that there was a moment where we sort of had to rethink the whole notion of how a genome translates into a lived life, specifically for humans, because, as you alluded to, genes don't do single things. They the stuff that Sarah was alluding to at the beginning, they do stuff at specific times and in specific tissues, and all genes are present in all cells. Or in all cells at some stage in their existence, and so you can't have them on all at the same time. They have to be switched off and regulated and turned on in these sort of cascades, these networks and circuits, in order that you can go from having all the information in every cell to only having the information that you need to become, a cell in the retina or a cell in your heart or a hair follicle or all of this.
So, Sarah, would it be fair to say that if you're writing a book, one of the most things that's beautiful about reading science books is how quickly they're out of date? That's one of the things. Is sometimes I go back to books that I had as a child and I go, this is wonderful. This is why it's beautiful. And that in terms of areas that if you're thinking, I don't want to keep rewriting this book, don't write about genetics for the time being. Because it does, I imagine the 21st century does seem to be that this is the century of a truly new understanding and a journey to a new understanding.
True. Because one of the books that influenced me a lot was the Human Genome in 23 chapters, or something like that, by Matt Ridley. And that, looking back at that now, 20 years later, roughly, it really is out of date. But it was interesting at the time. So what you say is absolutely true.
So, sorry, Sarah.
Yeah. No, I mean there'll be some truths, but things do change, things move on, and it's an incredibly exciting field to be in because things are changing so rapidly at the moment. And it's
In terms of people like me who in no way have any area of specialization in anything whatsoever, what are the things that people should be most aware of in terms of the changes in the last 2 decades? Because many people have on their shelves, they'll have books and they won't realize that. I mean, one of the things that I remember a lot of people talking about was the change in understanding, for instance, of what was, of junk DNA.
There have been a lot of revolutions since the sequencing of the human genome 20 years ago. And following on from the human genome, there was human genetic diversity, which we've been alluding to, which in some ways it's been, there's been awareness since the Talmud, but understanding that in a quantitative manner and comprehensively, both in terms of the genes and the non-coding regions, junk and regulatory regions and so on. That's really changed over the last 20 years, I think, completely. The understanding of somatic mutations, of mutations in tumors, cancer genomics, that's really changed over the last 10, 15 years. With much better understanding of the mutational processes, the different types of tumors. So that's genomics has provided a tool that's allowed an access to these different areas of basic science and applied sort of genomic medicine in incredibly new and powerful ways. And And then, of course, my area is gene expression genomics. So basically, how, where, when are the genes expressed? How are they regulated? How do cells transform along a developmental or differentiation pathway? What are the cell states? And again, it's been technology developments in genomics that have allowed that to flourish into the field that it is today. And now we're moving into spatial genomics, where we can actually sequence, in a very dense way, a tissue section, sort of almost down to single cell resolution. And so and that's giving us new insights into the microenvironments in tissues and how cells relate to each other, in incredible detail, which is throwing open new understanding of actually the structure and architecture of human tissues with genomic technologies, with the human genome sequence itself kind of being the foundation for the human work. But there is also the area, as you mentioned, kind of at the very beginning, about other genomes and how the human genome relates to, our primate relatives, non-human primate, and mammals, and then other sections of the tree of life. And so that, I think that's been incredibly, that's been completely transformed as well by genomics and, initially the Human Genome Project and then genomics more generally. So, yeah, so I think that there are many, many different areas that have sort of been ripped open and transformed over the last 20 years. So, and yeah, so our knowledge is changing. The textbooks being rewritten, and it is incredibly exciting.
So generally, what we would say is, if you have a book about which is called Your Guide to Genetics, and the date in the beginning is 1997, you need to update. You need to find a new one. Gil, I wanted to ask you about this. Is in terms of our understanding of nature and nurture, which of course I always know it's a very, increasingly, it's that beautiful bit of science where it seems to get more blurred. That there was a point, certainly in some conversations, I think in the media, which was this idea that genes were our destiny, and now it seems to be an understanding that they are a probability, but there are and how are we able to understand the differing potentials that we see when we are able to begin to understand genes?
Big question. So humans vary a lot, and they vary in many different ways. Some of those ways are very sort of striking early onset diseases that some people unfortunately have. Other way, and most people don't. Others are sort of propensity to get complex diseases like cardiovascular disease or Alzheimer's. Some are to do with behavioral traits, like whether you like to work in the morning or the evening. So people vary in many different ways. And what we've learned, I think, from the amazing amount of research that's gone into the genetic basis of human phenotypes and human diseases is essentially that genetics contributes to everything. There is a genetic component to, whether you like nodding on a Zoom call or not. Pointing at the person who's nodding in the corner. So genetics contributes to everything. It can contribute, though, in many, many different ways, some of which are very direct, some of which are very indirect and complicated. So the most direct ones are, of course, where, like hemophilia or sickle cell disease, where there's a particular type of mutation which affects a molecular trait, a cellular trait that ultimately gives you disease. And there's basically the disease is 100% genetics, and there's almost no environmental influence on that at all. But those are many of the rare diseases are like that. What we call Mendelian diseases, which have very striking patterns of inheritance within families, and that's how they were picked up and ultimately how the genes were found. But the vast majority of traits are really the result of a much more complex interplay between genetics and environment and exposure and history. And one of the great advances, I'd say, since the completion of the human genome was the development of technologies and experimental designs which allowed us to really probe into the genetic basis of these traits. So people have been studying things like heritability and how much genetic contributes to traits and disease for a long period of time, and they've known that for many diseases or many traits, genetics is 50% or so responsible. But what we've come to understand, I think, since our ability to probe the genome is really how the architecture is really very, very spread across the genome. So if you take cardiovascular disease, for example, there's not one trait, there's not one gene that increases your risk. There are thousands and thousands of places in your genome which can increase your risk. Now, some of those might be very direct because, for example, those are variants in some of the enzymes that process cholesterol, and therefore, if you've got a slightly wonky version of this gene, you tend to have high cholesterol, and we know cholesterol is bad for heart disease. But some of those might be very subtle, like they influence whether or not you enjoy smoking, or whether or not you really just can't bother to go for a run in the morning. So those do influence your risk of cardiovascular disease, but they're doing it moderately indirectly. And then there's one of the huge, I think, very cool findings in the last few years has been about how the genes that are not even in you can influence your risk of disease, but they're not in you, but they're in your parents or your siblings, because you're in an environment, the environment influences your risk of disease. There are a lot of influences on that environment. They're not random, these things, and they're influenced by our parents and our siblings, and they carry genes as well, and those genes will influence how they create those environments and the environments they make for us. And so you get this sort of second order contribution of genetic risk to a disease, which it is genetic, but it's not even going through me. So I think for me, that's one of the most amazing things that the genome has allowed us to do, is to sort of see all these different layers between a disease or a human trait and the very broad range of, and very often very subtle, genetic influences.
Adam.
Yeah, so just following on from what Gil was saying there, which is absolutely spot on. It's even more striking with the emergence of what I think is going to be a very exciting field in the last couple of years, and this is largely coming out of Sarah's campus, so at Shambles, and not actually Sanger, and it's social genetic influence. So. Now, I have to stress that so far this is in mice, and I don't think it's been done in humans yet. But the actual effect of the genetics, the genome, the genotype of a social interaction can be more significant than the genome that you actually bear yourself. Now, that seems like an absolutely nutty thing to say the first time I heard that, saw that as a result in a paper. Amelie Bird is the lead author on that. She's at Shambles. And I thought, well, that's completely bonkers. You're saying there's a bigger genetic influence on your behavior from genes that you're not even related to, but are part of your social network. And I suppose, I remember talking to her about this a couple of years ago, and we were trying to speculate about what this might, try to give an example for humans. And I think one of the ones that works quite well is that, so for example, being a night owl or an early riser has a significant genetic contribution to it. So if you're someone who stays up till 2 o'clock in the morning, there's a significant genetic influence to that, or vice versa if you like getting up at 6:30 and going for a run, as Gil said. Now, if you're one and your partner is the other, then it's quite possible that you will modify your behavior in order to align itself with your partner's behavior, which is the opposite phenotype, right? So you might, try your best to be a late riser if your partner is a late riser or night owl, or whichever way around it is. And so in that example, potentially, because this is a speculative example, it may be that the genetics of someone else is more significant in the phenotype that you are displaying than your own genetics. Now, we're just beginning to explore that in humans. In mice, you see it in many different characteristics to do with wound healing and licking behavior and nursing behavior, and the impact seems to be quite striking. I think we will see how significant it is in humans in the next few years as those studies get done. But I think it's just yet another example of, well, two things. One, as Sarah alluded to, we're just at the beginning of this, exploring the genome is going to go on for, all of us are going to be employed for a long time yet. And the second thing is the complexities of understanding our genome is only part of understanding our place in the environment, and now understanding our genome's relationship with other genomes that don't even have any sort of biochemical interaction with each other, but still have an effect on our behavior. I mean, it's kind of nuts, but that's what makes it so cool.
One day we'll do a phrase, I think you'll find it's more complex than that, is not required. But that never happens, does it? There's no easy. This is why, apart from cosmology, cosmology seems so simple when you just deal with holographic principles on the surfaces of black holes. Sarah, I wanted to ask about some of the kind of more, well, the specific work you do, which I now worry, because I remember once talking with Adam where he said more often than not when they say humans, what the research paper really means is mice. So I'm now worried that when I say, the human cell atlas, and you go, well, to be fair, it's a mouse. But your work on the human cell atlas, which it just, that again, in terms of the magnitude of the types, can you tell us a little bit about the starting point of this and what your ambitions are in terms of what you hope to discover with the Human Cell Atlas?
Sure. So the starting point was the resolution revolution in genomics, which is basically the kind of explosion in single-cell genomics technologies that have happened over the past decade or so. And that basically means that we're now able to measure the nucleic acid content of individual cells or even nuclei in a massively high throughput parallelized manner, with dropping costs. And that's what that sort of revolution in genomics, to be able to measure DNA or RNA in single cells, is really what gave a huge boost to this whole movement. And so that's gone hand in hand with incredible technology developments in spatial genomics and high throughput, massively parallel imaging methods, in situ sequencing, and so on, so that we can also study cells within their tissue context. And when I say tissue context, I am talking about human tissues. So absolutely, the Human Cell Atlas is about human cells and tissues. And that obviously begs the question of where you're probably thinking, well, where do you get the human cells and tissues from? And it's basically 3 different types of sources. It's either biopsies from living patients, so tiny little bits of tissue, a millimeter or a couple of millimeters cubed, or it's resection material from operations, basically discarded tissue from operations, from often tissue that's a normal, that's adjacent to a tumor, for instance, or a diseased section of an organ, or it's tissue from deceased transplant donors. And these are, it sounds strange, but it's actually very fresh tissue because they're essentially biochemically still alive, because they've just been extubated when the explants take place. And so there are those 3 different types of tissue sources, generally. Now the technologies are allowing studies of human tissue that have been preserved for long periods of time. So there's also things like FFP, tissue from biobanks that have been preserved and formalin fixed, that we use for the project. So that's a little bit of context. It is a hugely ambitious project. I mean, looking back, I sort of think like, what were we thinking 5-6 years ago? But the ambition is to really cover all of the 50 tissues in our body and to map them and chart them and understand them at that level of cellular detail, at mapping at the comprehensively using genomics technologies. And so what that means is basically gaining an understanding of all the different cell types and cell states of the tissues and organs in our body, understanding how they sit next to each other in their microenvironments in the tissues, and also how they're mapped within the bigger structure of organs. So you can kind of think of it in a hierarchical way, where you've got your entire organ, your heart, your liver, your kidney, and then you've got your different tissue microenvironments. In the heart, we've obviously got 4 chambers and valves and septum and vessels and so on, and then ultimately you're drilling down to the cells and the little microenvironment that they sit in terms of how the cardiomyocytes or epithelial cells sit next to vessels and immune cells and fibroblasts, stromal cells, et cetera, et cetera. So it's hugely ambitious. You're absolutely right. What's incredibly but what's very exciting is that it is a global community effort. And so that's something that's very special about this Genomics project, I think, compared to previous consortia, is that it's, we now have more than 2,000 members from almost 75 countries around the world that are participating, contributing data, developing technologies, developing computational methods. There are many, many different funders that contribute small or large amounts. So it's also very kind of grassroots and distributed in that sense. And the coordination, again, takes place in a distributed manner. There's a lot of sort of bringing together that Aviv Regev, who's my partner in crime and has been from the very beginning, she is at the Broad and is just about to move to Genentech. And we do a lot of coordination, but with the help of many, many different people who chair working groups focused on technologies or on particular organs and tissues, systems like the immune system and so on. So it's it's a community-driven effort, and all the coordination is essentially sort of voluntary coordination amongst the scientists. And so that actually works. It turns out that works. There are many millions of cells from thousands of samples, different donors, from over a dozen different organs in the data coordination platform that's publicly available at data.humancellatlas.org, for everybody to browse. The project is really, it's been going for less than 4 years, so it's really in the very early phases, and that's from the inception at the very beginning, kind of the first meeting in London in October 2016. But we're now already at the point where, there's a kind of rough, very, very rough scaffold of cells from different tissues, and they're now increasingly even being put into their spatial context. So the progress has been quite astonishing.
I love again, before I was bigging up cosmology, but the trouble is sometimes it's very hard to picture things at Planck time, etc. Whereas when I watch some of your lectures and when you're talking about topography and you're talking about the visual element of understanding, you're saying the scaffolding and the structure, I think is there are kind of Damascene moments in terms of understanding ourselves and living things on Earth. Gil, I wanted to ask you about your research, because I know that some of what I saw your great TED lecture, by the way, which is 1,000 genomes, 1,000 stories, and I think it was 1,000. I may well have done that again by a factor of 10. I seem to be having a habit of doing that at the moment.
That's the correct number. Well
thank heavens. But what I really want to talk about before we run out of time is the how you are using this new understanding and this incredible speed. You basically say that you can now you can map a genome overnight. So an individual for is it £3,000 approximately? You overnight you can map out their genome.
Yes, it's even cheaper than that now. So technology has really been the partner in crime with genomics. So many of the revolutions that have made the science possible have been driven by amazing sort of engineering advances that have enabled us to either sequence DNA or pull out single cells or amplify tiny amounts of DNA. Really the engine of it has been the DNA sequencing technologies, which are just amazing. The throughput and the cost has dropped enormously. So the first human genome costs, I'm going to get the number wrong, but it's $1 billion-ish, I think, to produce 3 billion. And today you can have your genome sequenced basically overnight. It depends a bit on the scale and so on, but for under 1,000, definitely. And what that's allowed us to do is to start to produce these foundational data sets that chart genomic variation at the truly populational scale. So in my field, I'm interested in understanding how genetic influences affect disease risk, and how we can use that information to either identify people who are at incredibly elevated risk for, say, cardiovascular disease or common cancers. I'm also interested in using it to try and understand human biology and use that to identify new opportunities for therapeutic intervention. So that's one type of population that we can now really probe with these technologies. But Sarah's populations of cells are another type of population. It used to be that genomics was a field where you did tiny little samples because of the cost of the experiments and their complexity. But now, there's this amazing project going on in the UK called the UK Biobank, which is currently sequencing the genomes of 500,000 people, and that's just amazing. In 20 years, we've gone from 1 to 1 million, basically. So that the important thing for me is that the scale is critical, because when you're trying to uncover patterns in biology, many of them are very subtle and complex. They don't sort of jump out at you. You need huge sample sizes and very rich data types on the scale of hundreds of thousands of observations to really see what's going on. And then you start seeing the patterns fall apart, and then you can integrate the sort of data you get in the biobank with the sort of data that Sarah's been generating to understand where disease risk lies, which tissues does it reside in, what are the cell types that are important, what are the molecular functions that are going wrong. And that's the sort of fuel that allows you to do things like identify new drug targets. So for me, the key thing is essentially it's a technology-driven ability to scale in a way that we couldn't before.
Adam, yes, if you'd like to. I've got a question that's just come in for you, but please add if you would like.
I was just going to add to that by saying that the trajectory of genomics from that from 20 years ago with the beginning of the Human Genome Project, or the first draft of the Human Genome Project based on one person, but even before that, when we were identifying individual genes, often for the common diseases or the Mendelian diseases, what we were finding is common variants. So things that they vary between people, but they can be identified relatively easily because there aren't that many differences. Now, I think what in the last 10 years, more than at any other time, and what a lot of the way that this is going, which is why people like Gil need very, very large data sets, is that what we're finding is that a lot of the heritability, or a lot of the disease risk or behavioral risks for a relationship between a genotype and a phenotype are actually hidden in rare variants, right? So things that vary between bits of genes and bits of non-coding DNA that vary between people at a very much lower frequency. And they're hard to find. They're hard to find because they're rare. And yet what we're finding more and more is that they are going to account for enormous amounts of the biology that we consider to be important or that we want to discover. And the only way you can get around that is by getting more genomes, by finding more rare variants, by looking at as many genomes as you can possibly get your hands on. I mean, I think there is a genuine case, and I talk about it in that book, that the same chapter that I showed that betting book. There's a genuine scientific case for sequencing everyone's DNA, for a full genome sequence from birth, as a standard, as part of the NHS. There's a lot of ethical concerns with that. There's a lot of data security concerns with that, and privacy concerns with that. And all of those conversations are happening and need to continue to happen. But from a scientific point of view, I think that a lot of us think that is how we're going to understand human evolution, human variants, behavioral genetics, disease genetics, cancers, the whole shebang, why we are the way we are.
Well, we will talk towards the end about ethics as well, so I'll just throw that out there now. But Adam, we've got a question which is for you. This is a question from AI, and I really genuinely hope it is some AI. I hope it's AI that's passed its Turing test. So AI, congratulations, and we will send the certificate. How do we improve our scientific language when we talk about race?
Well, that's a great question. That's a great question, and I wish I knew the answer to that in a definitive way. So one of the things I talk about a lot is human variation in relation to the social construct of race, which is one of the things that the Human Genome Project, more than anything else, has shown, is that race is a social idea. It is a consensus, the way we talk about race, rather than a biologically encoded, meaning genetically encoded, classification system. And the truth is we don't really have the language to talk about the complexities of genomics at all. And there's a sort of deep stem to this, which is we don't really have the language to talk about data sets of the human genome. Is the most complex data set that we have ever come across so far, and will be like that for a long time yet. We don't really teach probability or risk, particularly probability in relation to genetics, at a school level. And the narrative of genetics is the thing that we were talking about at the beginning of this show, which is one gene for one thing. You've got a gene for blue eyes or brown eyes, which seems that's still in the syllabus, right? It's sort of true some of the time, and when I teach GCSE or A-level students, I feel I have to tell them, answer what the textbook says, because you're not going to get the marks if you say what is actually what we know, which is that it's partly true some of the time. But that's genetics. That's the reality of genetics. This stuff is wickedly complex. And so when it comes to issues like race, which have a very political and very sensitive aspect to them, especially given that our understanding of race has a historical trajectory which is absolutely rooted with the birth of science, subsequently the emergence of anthropology, from which you get the emergence of genetics, only 100 and so years ago. And we're only a few decades out of that era. And we've got some definitive answers in there, but taxonomizing things is so culturally, it's so part of the human condition. And one of the things I say in my lectures quite a lot is that, evolution, particularly as expressed in genomics, in the human genome, has unfolded over 4 billion years and never had any intention of being deciphered, right? It had no foresight there. And so the reason these guys have got a lifetime's work ahead of them is because there's really no structure to it. We don't really have an agreed definition of what a gene is. And yet we've got, hundreds of thousands of geneticists all around the world talking about genes every single day. And that's the thin end of the wedge. Anyway, the question wasn't about that. So the answer is, I don't know. We talk about populations, we talk about ethnicities, we talk about ancestral populations. In our databases, such as UK Biobank, we have problems which are effectively aspects of structural racism, because we know much more about the genomes of European people than we do about the genomes of anyone else on Earth, particularly people from Africa, for whom their genomes have more diversity in them than everyone else in the rest of the world put together. So there are these massive problems. We don't quite, you still read papers by geneticists which say, which talk about, extrapolate data when actually what they mean is white Europeans, or talk about Caucasian populations, which is a sort of meaningless, weird term that needs to be absolutely abandoned, possibly decades ago. But I don't know what we replace that language with. And that's an active conversation that we're having in genetics at the moment. So it's a great question, Al. And the answer is we don't really know, but we're thinking about it very hard.
Thanks. I think one of the great books about looking not specifically about genetics, but in terms of looking at that, the desire to go, this measurement, I want it to fit exactly with what I believe, is Stephen Jay Gould's book, Mismeasure of Man, which was about the desire to find very specific differences in brain size. And it's a very good book looking at the kind of the highly flawed research methods that we used. Sarah, what are you, in terms of, we were talking about ethics there, and do you have, within your areas of research and around that, are there any areas you think we really need to be talking more and more about what the ethics are and what our understanding is, and how sometimes, as we know with any interesting science, often in the media they'll read one sentence of that paper and they'll publish that. So what is there anything you're particularly wary of in terms of misunderstanding?
Well, I agree with Adam that that there are vast differences in I'll say the density of research in different types of human populations or human samples, and that's one of the things that we have an eye on, and we're sort of actively trying to steer against in the human cells community, and that is to include to try to include as much genetic diversity as possible in our samples, and we're very lucky that there are funders who funded Human Cells Project focused precisely on that, basically covering genetic diversity in different contexts. Profiling, using single-cell genomics to profile blood samples from populations around the world or populations in one city, of different genetic backgrounds, samples of different genetic backgrounds, donors of different genetic backgrounds, and so on. So, I mean, that's one thing that's talked about, and we're very conscious of, precisely because of the bias that quite frankly, biomedical research, I don't think it's just genetics, it's biomedical research in general has focused more on some populations than others. And then the second thing, which is different, is also inclusion of scientists, a diversity of scientists, both, again, kind of global geographic inclusion, of scientists engaging different communities, but also, being aware of inclusion, even in Europe, UK, North America, and so on. And I just had an email earlier about, for instance, Black women in computational biology, a kind of special interest group, which is just a fantastic community that's basically supporting each other, and which many of us are allied to, and so we're trying to strengthen our community by including diverse scientists and diverse samples, and we think that will make the community, the scientific community, and the scientific project much stronger and of higher quality, basically.
Gil, what do you think are in terms of the major ethical issues?
I agree that equity is probably one of the most important ethical issues, in that we want the advances in medicine and healthcare that come from our ever increasing genomic understanding to be shared equally by everyone. But as Adam pointed out, that research is not well distributed across the world, and there are very good reasons why we think that doing research in one particular type of locality may not translate 100% into other localities. So equity is a very important one. Other people have touched on that, so I don't want to spend lots more time on it. I think the other big, it's an ethical issue, is around data and data sharing, data access. The world we work in is only made possible by our ability to access very rich data that links the genomic information on very large numbers of individuals to very rich information on their health and their disease and their treatment, their outcome. And so what you worry about, what I worry about, is the sort of data protectionism that can creep in, like you can't access my data, it's special. One of the things that's made genetics as a field really stand out, I think, is its the way it has not fallen into that trap. Certainly, it was driven very much by a culture of openness and access. Projects like the 1,000 Genomes Project or the Human Cell Atlas or the Human Genome Project originally, they've all effectively been open data projects, which has been fantastic. And I think one of the things that we need to have greater clarity on is, as we start putting more and more information onto a genome, like my entire healthcare records and what I had for breakfast, what's the right way of thinking about who should get access to those data and under what terms? Should they pay for it? Should they get it for free? What's my right? What are my rights within that? And some of that, I think, we can take good principles from things like GDPR and so on, but I think there's a much wider societal question about what rights does society essentially have around accessing your data on this kind of scale in order to drive this type of research. So that's the other thing I'd identify.
We're near the end now, so we've actually managed to get through the whole thing without talking about COVID-19. So, which is, but I will briefly. But we don't have to talk about this. No, Adam. No. Adam has done some very interesting stuff on that, which I highly recommend you look up, and you will see both writing and broadcast. You can talk about this to an extent, which I'd like to ask all of you in terms of your ambitions for when we are facing battles in human health. And I wondered what, the hopes of current research in genetics, in genomics, where you hope we will be, and what you hope are going to be the possible breakthroughs. So, Adam, can I start with you? On that?
Yeah, sure. And I sort of want to pick up from something that Gil was just saying there as well, which is that something which I don't think anyone really anticipated happening in the last 20 or 30 years was the enormous commercial potential for monetizing people's genomes, which has occurred through primarily for companies that are expressing interest in ancestry, genetic genealogy, and these are 23andMe or Ancestry.com, and I don't, I'm not in the business of telling people how to spend their money. I think that the tests themselves are of trivial interest. The results are mostly of trivial interest. And they also do something which is, I think, we've been arguing against or trying to teach against for the last 30 odd years, which is that kind of genetic determinism that which is not the way geneticists think about the relationship between genes and behavior these days. So I'm sort of generally opposed to them, whilst recognizing that they have some sort of trivial value to them. But what I think is more significant is that companies specifically, such as 23andMe, their fundamental interest is not in ancestry; it is in commercializing people's genomes. And the amazing trick that they have successfully pulled off is getting people to pay to give their most private data to a company who subsequently are going to monetize it by selling it on to pharmaceutical companies in order to develop drugs, which subsequently will be sold back to them. Now, I don't want to sound like I'm sort of hugely politically motivated by saying this. I sort of think it's astonishing because when people buy those kits, they're not the client, they are the product. And you can opt out. And I suggest that if you are interested in taking an ancestry test, and just caveat emptor your way through this and make sure you know what you're getting into. My main problem with that whole process is not the generation of new science or potentially new drugs that will help people. It is that all of this is happening behind closed doors, right? And the whole of the Human Genome Project, Sarah's she's caveating that. I think she's right too, but because there are commercial partnerships with the public sector as well. But the whole Human Genome Project was set up as an open data exercise, the free information, the free exchange of information by default in perpetuity. And I think that science progresses best when there is a full free flow of information, which genomics has done so well for so many decades. It bugs me now that the largest data sets for human genomes in the world are not in UK Biobank or the 100,000 Genomes Project or Genomics UK or any of the publicly owned databases, but they're owned by 23andMe and Ancestry.com. I think that is suboptimal for the progress of science and therefore the understanding of how humans work.
Sarah,
so you know, you're right to sort of caveat, absolutely. I'd say it's to me it's you know, they're not complete genomes, and the taxpayer, I think, or even large philanthropic funders are probably still bigger funders than the private sector in terms of being able to be a motor of biomedical research and fund open science and so on. So there's also, you know, glass half empty, glass half full. I'd sort of put things in perspective. The other point that I'd like to pick up on is that it's not just the genetic domain. I think we're only slowly waking up to privacy issues. So we're also the product of our data, you know, in social media, in shopping, even, you know, physical shopping or online shopping. We're giving away our data all the time and not doing those opt-outs because they take too much time or they're not explicit and so on and so forth. So I think it's only very, very slowly that we are, as a society, we're realizing the value of the data about ourselves in all different domains. It's not just the genetics. You know, this is kind of, you know, we're giving away something about ourselves in this very program, in the, you know, the pictures of our houses and so on, and where we are at this very point in time. Right. Well, you laugh, but that's you know, that's part of it. And so it's so I think we have to become more aware and have a conversation about how to deal with that, and what is it that we actually want, because maybe we want to give away our data to the company because we think it's a good cause. You know, some people may feel that way. Most people probably haven't thought it through, but there may genuinely be some people who think that. There may be others who want to charge money, and you know, the people we have to be more explicit and give and have pathways for having those options, rather than there just not being any option. But that's something that's the society as a whole, I think, is sort of living through that conversation. I mean, in terms of you know, sort of leaps of science in genomics, I think there's the basic genome sequencing of every person, of course, you know, and personally, I think that will have the biggest impact in pediatric and neonatal medicine. You know, and the diseases that penetrate extremely quickly after birth and are sort of devastating Mendelian disorders and need to be diagnosed quickly, you know, and then slowly, possibly throughout life. And you know, what I see sort of going forward is of course, cell atlas technology is becoming part of mainstream medicine. just the way genome sequencing has, over the last 20 years. So we would be, single cell sequencing blood samples, tissue samples, sequencing tissue sections of biopsies, and basically being able to diagnose diseases in detail in ways that we've never been before, be able to engineer tissues for regenerative medicine based on those templates that we have now of in vivo tissues, and be able to patch and fix broken organs and tissues by simply making copies that are based on the in vivo templates. So those are the kinds of things that I'm looking forward to in my lifetime, let's say 20 years from now.
Gil, you in terms of your work, in terms of targeting drugs, etc., but what are you hoping we're getting close to in terms of breakthroughs in this area?
I think the area what I want to see the most, and Sarah alluded to there, is essentially being able to integrate genomic information genuinely into healthcare. I actually don't think we've done it at all, really. We sort of scratched the surface with genomic medicine programs, but that's really focused on people who've got disease and the diagnostic problem. I think that genetics is now at a point where the predictive information about risk that you can get by looking at someone's genome is a useful thing to know. We can identify women at risk of breast cancer, at an incredibly elevated risk of breast cancer, not because they've got one of these really strong familial diseases, but they just happen to have got really unlucky in the thousands of small effect variants that influence your risk of getting that disease. That information, I think, should be being used today within healthcare. We know how to sort of bring that into screening programs. We can do similar things in cardiovascular disease. And there are lots of other diseases where that information now genuinely can be used in a preventative way rather than a responsive way. So that's the thing I really want to see. But it's part of this bigger story, I think, of translating, the research that was made possible by the sequencing of the genome right through to something that will impact individuals' lives.
Brilliant. Thank you all very much to Sarah, Gil and Adam. And I think under this as well, at some point, we'll try and put up some links as well to other lectures that they've done, so you can find out more about the work and more about the things that they're doing. Thank you very much to the Genetics side of the Milner Centre for Evolution. Adam, it's been very interesting because there's just enough lighting on the landing to know that some of your family have got the night owl gene for sure. I don't think they followed your pre-show instructions of going to bed. And don't forget the Sunday Science Q&A on Cosmic Shambles. That we do that every 3 p.m. every Sunday, 3 p.m. that's in terms of British summertime. And thank you very much for joining us. Thank you very much to Trent Burton for producing this, and we'll be back with another Genetic Shambles in 2 weeks' time. Thank you.
Thank you. Bye.